Today we will discuss the detailed anatomy of cornea. Let's know about the origin of cornea. The word cornea has come from kerato. The term kerato in Greek means horn or shield-like. Ancient Greek used to believe that cornea is derived from same material like that of thinly sliced horn of animal. Introduction. Cornea is avascular and transparent, accounts for 70% of total refractive power of the eye. Principal optical surface of the eye. Dimensions of cornea. Anterior surface is elliptical and posterior surface is circular. Diameter. Horizontal diameter 11.75 mm and vertical diameter 11 mm. Posterior surface diameter 11.5 mm. Radius of curvature. The anterior and posterior radius of curvature are 7.8 mm and 6.5 mm. Thickness. The central corneal thickness is 0.52 mm, whereas periphery is 0.67 mm thick. Central corneal thickness has direct influence in IOP measurement. The central 5 mm of the cornea forms the most powerful refracting surface. Histology. Cornea has 6 layers, that is anterior epithelium, Bowman's membrane, central stroma, Duar's layer, Descemet's membrane, endothelium. Duar's layer also called the predescemet's membrane. Let's know about the corneal epithelium. It's 50 to 90 micron thick, stratified squamous type, consists of 5 to 6 layers. Corneal epithelium seeds at regular interval. It takes normally 7 days for replacement. Entire corneal epithelium. Tight junctions in basal cells account for epithelium's transparency. Bowman's membrane binds the corneal stroma anteriorly with basement membrane of epithelium, not a true elastic membrane. Once destroyed, does not regenerate. Shows considerable resistance of infections and injury. Stroma 0.5 mm thick. The stroma or substania propria occupies about 90% of the total corneal thickness, composed of collagen fibrils, keratocytes, and extracellular ground substances. The lamellae are arranged in many layers, 200 to 250, and are parallel to each other. Anterior one-third stroma. Lamellae are thin. It's like 0.5 to 30 micron wide and 0.2 to 1.2 micron thick. Posterior two-third stroma. Lamellae are larger, 100 to 200 micron wide and 1 to 2.5 micron thick. Duar's layer. It was discovered by Dr. Harminder Singh Dua at the University of Northam in 2013. It is the fourth layer, 15 microns thick, strong layer and impervious to air. Descemet's membrane, a strong homogeneous layer, basement's membrane of endothelium made up of collagen and glycoprotein, elastic. It can regenerate resistance to chemical agents, infection and pathological process. On posterior surface, it shows hazel-Henle bodies which increase with advancing age. Endothelium. Single layer of flat polygonal cells. At birth, cell density is around 6,000 cells per square millimeter. With increasing age, the number of cells is reduced. Involved in active secretion and protein synthesis. Contains highest number of mitochondria next to photoreceptors. Let's know about the blood supply of cornea. Cornea is an avascular structure. Small loops derived from the anterior ciliary vessels invade its periphery for about 1 mm and provide nourishment. These loops are not in cornea but in the subconjunctival tissue which overlaps the cornea. Nerve supply. The cornea is one of the most sensitive tissues of body and has one of the richest sensory nerve supplies in the body. It is supplied by long and short ciliary nerves from the ophthalmic division of trigeminal nerve. Most are concentrated in the anterior stroma beneath the Bowman zone and send branches forward into epithelium. Descemet's membrane and endothelium are not innervated. Junction of cornea and sclera contains trabecular meshwork and canal of scheme. At limbus, corneal epithelium becomes bulbar conjunctival epithelium. Bowman's membrane becomes continuous with the lamina propria of the conjunctiva and Tenon's capsule. Stroma becomes sclera. Descemet's membrane becomes Schwab's line. Endothelium lines the trabecular meshwork and becomes continuous with the anterior surface of the epithelium. Corneal transparency. Let's know about the corneal transparency. Tight junctions of the epithelium cells, endothelial pump machines, absence of blood vessels, absence of pigments, sacricity of cells nuclei in stroma, uniform structure of stroma.